nice introduction. So today I'm going to talk, already Holden mentioned that I'm going to talk about the role of bioinformatics in the immunomics technologies. And so immunoinformatics is fairly a new term in the field of immunology, but it's, I must say that it's rapidly evolving and also growing at a very fast pace. So I'll just start with the basics of what we mean by immunoinformatics, and it's actually an discipline of bioinformatics which utilizes or is a cross-disciplinary area of the mathematics, information science, computer engineering, genomics, proteomics, and how we integrate all this together to bridge the immunology and informatics. So the bioinformatics area enables you to do data-driven research strategies and systems approaches to understand the networks which are regulating the immune system. And considering the breadth of the topic, now immunoinformatics was composed of a cross-section of research ranging, as I mentioned before, data integration. It also includes how you could do the epitope predictions to the systems level applications. So today what I'm going to show is the efforts that's been going on here at Stanford. And it's going to be an overview because there's a lot to cover. So I'll go with the basics first. So what are the main immune-related threats? Like, you know, the pathogens, autoimmunity, organ transplantation. So where we do see the immune system plays an important role. And okay, thank you. So I will show some of these examples from these areas. So recently, a paper came out in Nature Immunology from Brian Kidd and Joel Dudley, and they were our colleagues here and now moved to Mount Sinai. And I really like the way they have depicted that how the immunology or immunoinformatics is going forward, but with the different kinds of modalities that we have. Like, for example, from a person, we capture the clinical data, you know, protein information, and then the cell informations on site of platforms, metabolomics data, transcriptomic and genomics. So we have tons of data for a single person and so on for a group. But how we can now integrate and understand this at the systems level, and so we need informatics, and so basically unifying the immunology with informatics and multi-scale biology. And what we want to get to is, like, let's understand the healthies first, the healthy population, and then go to the disease. So some of the data that I'm going to show is, or what we are trying to attempt, is to understand first the normal populations and as well as disease. But we really need to understand the normals first before we try to understand the disease individuals. So I'm sure in last 2-3 days you had been hearing a lot about high throughput technologies that's been done here at HIMC, and primarily I just listed 3 of them, but there are many more. Like Luminex is used for cytokine profiling, CyTOF is a molecular flow cytometry, mass cytometry technique, and then fluidigm biomarkers for the qPCR arrays, where you can quantify the genes at the single cell level. So Luminex, I'm not going to go deep into it because I'm sure Yale has covered a lot on that. But this is a multi-bead assays, and you can quantify about 50 cytokines in parallel from any given sample. Like that, you have multicolor flow cyto, the CyTOF technique, which uses the antibodies which are labeled with heavy metal ions. So the biggest advantage over the conventional flow cytometry method is there is no spillovers, and you can detect at the most 60 plus parameters. And so that is, again, that's telling you that what we are getting is lots of data from this. So how we analyze that. But before I go into it, it's a fluidigm biomark is another technique where we quantify the genes from a sorted population. So you can sort the T cells or B cells and look at the gene expression for the single cells. So So given that I have just gone over very quick with these high throughput assays, but I would like you to go over these buzzwords here whenever you are dealing with high throughput data, and that's like experimental design is very, very critical, and I'll show you why. And then you deal with a lot of batch effects. Then normalization is also very important when you are looking at this high throughput data. Then and then coming out with the statistical approaches to analyze the data. You need a database because you are not just dealing with Excel spreadsheet anymore. These are lots of data and coming out of this techniques, right? So it really helps if you have this data in a database format, and then the software packages. And I'm a believer of more of the open source ones, but you could use the commercial ones. So the first point that I want to touch today is experimental design. This is absolutely critical to determine the batch effects, and this is based on my experience where the immunologists, they come to us, and they by the time they come to us, they come with all this data and say, what do I do with this data? But then I always ask them, so how is your design? And they say, oh, we just designed the way we wanted it to be, but that's not the case. I will recommend that please, or suggest that please go to the statisticians before you even start your experiment, because it is absolutely very, very important that how you design your experiments, because down the line when you are doing the downstream analysis, these factors these are the confounding factors which adds up to your results. So here is an example where on your right hand side you see an experimental design where in one, let's say these are like you're doing it in plates and in batches. So in one plate you have all the controls. In another plate you have all the disease groups, right? And then here is the one where it's mixed. So now if I want to compare all these batches together, I mean, it's impossible, because we already by now have introduced a lot of biases, because this is based on like in one batch you have controls and the disease. So how do I compare? How do I normalize them? But on the other side, if you look at this one, where if you put mixed samples on a plate, it's much better way of looking at or finding out the error. So you can estimate the error based on the batches. So what I mean to say here is that there are a lot of confounding factors like laboratory conditions when you are running it, and since you have lots of samples, you cannot run it in one single day. So you need to distribute them into batches, and there are like reagent lots are different, personal differences. So batch effects really lead to increased variability, and it decreases the power to detect a biological signal. So for normalization purpose, I mean, always, I would recommend that if you can design in this fashion, that helps you. So the message was just that be careful about your experimental design. So how and where does bioinformatics come into all of this? So I'm coming from a place where it's one gene, one PhD. But now it's like you have like 50,000 genes to look at, and not only just 50,000, you have so many other data types that add to it, right? So from qualitative to quantitative, and from descriptive to we are moving more towards a predictive analysis. So from biological measurements that we get, we need to have some kind of a computational model. We need to take help of this computational model to get to the biology. So one of these efforts that's been done in Mark Davis's group, and initially I got involved in this one, where we are looking at the one-stop shop for a multi-level biological analysis of blood from individuals of different age groups. So which was done was that the blood was collected from the individuals. There were 90 patients, and then the cells were sorted for flow cytometry, and then for immune cell stimulation, phosphoflow technique was used, and then to measure the cytokines, the Luminex technology, and then for whole blood gene expression, the microarrays were done. So as you see that so many different kinds of data was obtained. So typically the workflow for this kind of data is that you start with the raw data, you do quality control, and then you normalize it, and based on these are the steps, and then you go for statistical testing and data visualization. And that's another key aspect, that how you visualize the data. For high throughput data, data visualization is very, very important. And some of them you might have seen, SPADE and a lot of other programs that have come out with excellent visualization for the high throughput cells data. So I'm not going to go over those a lot here, because already other people have explained it. But the first thing that you do when you get a raw data is you ask this question of reproducibility. So reproducibility, how much is the variation, which is an interesting, a biological variation of interest, and the one which is coming out of the system. So that's more of noise. So you need to separate them out and also look at outlier detection. This is a way, like, you're just not looking at just 2D plots of just, or a scatter plot of a single data. When you have tons of data, this is one of these like matrix approach where you look at the data when you, and comparing one patient to the other, depending on what the samples are. But these are like, these are scatter plots, but you see everything in one view and get some of them which are like outliers. So what this means is, like, you can see all the data at once and then find out and detect where you see the outliers, and then remove those samples from your analysis. The other way of looking at the data is more, these are called the bean plots, where this is the data from flow cytometry data, looking at the cell subsets frequencies of the same cohort that I just mentioned before, where we had 90 patients. And what we see here is that on an x-axis is the young, mid-age, so the patients were distributed based on age. And then we're looking at the hemopoietic lineage, the PBMC subsets. And so we start with lymphocytes, monocytes, CD3, CD4, and CD4 subsets, and on this side is all the CD8 subsets. So if I zoom in and see one of them, you can see that definitely age, for monocytes, age has an influence. So again, like, just don't look at one single data point, but bring everything together to have the whole picture. The other aspect is the principal component analysis. That's another, when you are working with high dimensional data, that's pretty common nowadays. When I say PCA, it's no fancy anymore. But principal component analysis is another way of looking at classifying the groups based on cytokine profiles. So you could see that when you bring in all this data, all data from the patients, and then cluster it together, this is more, they separate out very well. So what I'm showing here are, based on their cytokine profile, how the young, the mid-age, or young or elderly patients are separating or segregating. This is a very commonly used dimension reduction technique. So I'll just, for this part, I just want to thank Holden and Mark and Atul and the other HIMC members who helped me getting the data, and I did some analysis. Now I will move on and talk about my new role in IMPORT. And I don't know how many of you heard about IMPORT, but IMPORT is a data warehouse for immunology data sets, and not only just research data from the institutes, but it's also, we are getting a lot of open source clinical trial data. And so it's open source, I call it as data warehouse, and also they have some limited tools to do like Flock, which is an auto-gating tool for flow cytometry data. So anyone can log in, register, and then can get access. So if you see here, right now, IMPORT has 89 studies and different kinds of assays, and right from ELISA, ELISpot, flow data, gene expression, HAI. So you can just go and look at it. What if some of these, there are a lot of diseases also in there, so you can download if you have some disease that you are working on and you want to compare the data. Then this is a source. And a lot of Stanford data from HIMSI consortiums also goes to IMPORT. So who are the users? Basically, the users is anyone, like I call it a scientific explorer, like a person who just wants to do a Google search. Then import has a search where you type in any cytokine or any cell data, and it gives you that it returns back the studies where they have the data of your interest. And then we have users like scientists who has a data, who is working in the lab, and he generates the data and he needs some tool. So when what you could do is that you can use Flock. Flock is an auto gating tool, and then Immunoexpresso, this was developed by Shai Chenor here, and that's more of a text mining tool, and a lot of work also been done on the ontology side on import. So basically, a scientist who comes in with the data can and wants to explore data sets from their area of interest, they can go here, or also a sophisticated user, meaning a person who has a lab data but has limited bioinformatics skills can look for these analysis tools available and to make sense of their data. And then the bioinformatics scientist, a person who is in a dry lab and who is interested for meta-analysis. This is an excellent source where you download all these data sets, and the import data sets are available in MySQL. So you can get a MySQL dump, and you can store it on your local server and query these data sets. Okay. So I will focus more on the category 3 user and for how we can do the meta-analysis. And one of the things that you could do is, this is just an outline diagram of how you take a data which is already, someone has analyzed it, submitted the data, but then you could also reanalyze. And so this gives you, the purpose of reanalysis is that you can establish the statistical significance with studies. Let's say that you have your own study, but then you need to have, you need some confirmation about that. So why don't you look at if there are studies available, you download them and you compare it, and so that gives you like an estimate of how the magnitude, and also it helps you to examine the subgroups within the individual numbers that are not statistically significant. Let's say you don't have much power. You started with 10 patients, but then if you have some data which is close to yours, then you bring in more data, and then that gives you power. So the advantages are like having greater statistical power, confirmatory data analysis, greater ability to extrapolate to general populations. So you look at the way I'm going to show you how we have done it for normal populations, and also consider as an evidence-based resource. On the other hand, I'm not saying it's all starry, like there are disadvantages as well. So it's difficult and time consuming to identify the appropriate studies. And not all studies provide an adequate data for inclusion and analysis, right? So we have to just take out what we need. That's what I would say. So today I'm going to show you my work on, and this is a case study where I have reanalyzed one of the data from a seasonal allergy. It's a clinical trial data. And so the aim was to understand the mechanistic understanding of omalizumab, which is widely known as Xolair, and allergy shots for the treatment of seasonal allergy. So this study was done for the ragweed allergies, and ragweed these are the flowering plants, and they cause allergic rhinitis, which are triggered by the airborne allergens such as pollen and dander. And the common symptoms that we all get every year is either stuffy nose and runny nose and so on, itchy eyes. So the mechanism for these allergic reactions are that it causes an excessive activation of mast cells and basophils, and the increase in, at the end, there's an increase in the IgE levels in the serum. So this study was done, and it started way back in you see here in 2004, and it ended in 2012. And this clinical trial was done, this is in collaboration with Novartis and Genentech, and so they submitted this data, and this data was the first one in import which came out as an open source clinical trial data. So we looked into it and say what we can do or what we can learn from this clinical trial data. So what we did is, before I go, let me give you some background that from this trial they published 2 papers. So one of the papers says that what they found was that omalizumab pretreatment enhances the safety of the ragweed immunotherapy for allergic rhinitis. And then they also looked at the combination treatment where the drug plus the immunotherapy was applied, and it shows that it has increased benefits. So these were the 2 papers which came out. We read the paper, and before I go into it, I'll quickly go to how the allergen works. So as soon as our system, the immune system, sees the allergen, it binds to the IgE antibodies through the FCE receptor, and then the mast cells get degranulated, and you see the symptoms of you see all these allergic symptoms. On the other hand, with this allergen-specific immunotherapy, if you give a little bit dose of these allergens before the peak season, then that's called priming dose, and that tells your system that there's an allergen, and you raise the antibodies against that allergen. And that's what is showing here, that an allergen then gets bind, and then the B cells then start producing the IgE from IgM. There is a class switch to IgE, and then in response to allergen, they produce a lot of free IgE. I'm not going to spend more time, but this is the basics, and just show you the mechanism of action for the omalizumab. So what omalizumab is a drug. Okay. So omalizumab is a drug which binds to IgE, and that way the IgE then cannot bind to these mast cells, because that causes these all the allergic reactions. On the other hand, if, like you can see here, this is the allergen which binds to IgE and then through the FCE receptors, right? But when the drug competes to IgE and doesn't allow the IgE to bind here, and that's what that's what's the role of omalizumab. So in short, when the system sees omalizumab, then there's a decrease in free IgE levels, and it also down regulates the expression of the FCE receptor on the mast cells and basophils and dendritic cells. So what data I have from Casale allergy clinical trial day? So there are 4 arms. So we started with placebo, then immunotherapy, where you get a little dose of these allergens. Omalizumab, that's the drug, and then omalizumab and immunotherapy, that's a combination. And then this is was the dose schedule for the study, and what we know from each patient. So what we know from each patient is their allergy severity score, which is the primary endpoint. From the serum, they collected all these IgG, IgM, allergen-specific IgG levels, and then they had a CBC counts for basophils, eosinophils, hemoglobin, and so on, and lymphocytes. On the other hand, on the other side, this is the race, gender, all the demographics info, and then the set of questionnaires about who had the allergy before and all the histories, clinical history. And along with it, this is all clinical data, but then they also did run the flow panels, where they had about 8 panels and to identify these cell populations and the gene transcript. So what I'm going to show you here is a tons of data. So when I look at it, I said, wow, this is a mine, because you have so much of data. But then we have challenges. The challenge is that how we integrate all this together. And this is all from open source, right? So the first challenge that I had is that this is not my study. So how I align it, how I understand it. So this took me some time, that how to align all these different kinds of data that they captured. So like, serum analyte data, the microarray flows, and the allergy severity score. So, and the units were different. In some data sets, I got the data collections in visits. the other one as a data collection study day, and then maybe in some other units. So to put it all together was a nightmare for me initially. But first, when I started understanding that how they have done, then it was a lot easier, because for any analysis, when you have like different kinds of data, you need to align them before you start asking the questions. So I was able to align the data, and then I moved on and said, okay, now I start asking the questions from this data. So I was trying to address the questions like, understanding the molecular or cellular perturbations which correlate with the primary efficacy, and also identify the responders and non-responders to the therapy. So this shows, that when you download any data sets and you have papers available, try to see if you can reproduce their results. So what I'm showing here is that combination therapy was most effective, where the allergy symptom goes down, and this is the peak allergy season, and for combination therapy, as I mentioned, went down compared to the other arms. And this is the paper from the this is a figure from the paper, and I was able to reproduce. So you need to do confirmatory analysis before you go and explore your own what you want to do with the data. So so I downloaded all the, so my interest was more on the flow cytometry data. So I downloaded all the flow data and then did the gating based on this auto gating tool called FLOC. And so it's the K-means algorithm approach. That's what they have used for isolating the group of cells based on the cell populations, and how this is how it works, where they have created a gradient where 1, 2, and 3, and 4, and based on these levels of expression, you can isolate what these populations are. So this is more of the visualization. On the other hand, this is the way we get the data, where you see the expressions of all these markers and the frequencies or the proportion of each cell, each population. So once when I did the run my flow data through FLOC, this is the output I got. So the first thing that being a bioinformatician I encountered was that, okay, how I define the cells. There are predefined cells, right, in the literature. So I started reading Holden's paper and looking at these, how he has defined the cell populations. But then at some point it became tedious for me that how I look at and read all the literature. So then I took this approach of combinatorial approach. So based on, let's say this is an example of, in one panel we had 4 markers, and then based on these FLOC output, they give you directions, and then the expression score. So 1 is low and 4 is high, 1 to 4 in the scale of 1 to 4. And then based on these markers, then I took this approach of doing the all possible combinations of these markers, direction, and the expression score. So this is just an example of how you combine all these markers together, and that yielded me around 55,000 cell populations based on these all possible permutation and combinations. I started with any 2 markers, and at the most the 4. And then I got to the cell population summary, and then filtered the cell populations based on if there is a variation greater than 40% in any one arm across all visits. So based on that, I got about 11,000 cell populations, and what I'm showing, this is exactly the approach I've taken, how you're familiar with the gene data, right? So when you have microarray data, you analyze it, and then on in rows are all the genes, and then columns are all your samples, right? So instead of just genes, now I have all cells, the cell populations that I've identified, and the columns are all my samples, and this was a longitudinal study. So these are the 4 arms and then different visits. So I started looking at the pattern, that how what I can get from here. So this was on a summarized data. So I summarized the data based on the cell based on the number of subjects in an arm and per visit. So this is not on individual data, but this is all a group summary. So then when I had all these cell populations, I didn't know where to go. So I started very simple queries as, okay, locate all the cell populations where the CD14 is greater than or equal to 3. That means higher expression of CD14 leads to the which is a monocyte cell marker. CD19 is the B cell. So I started locating them where these cells are. So let me zoom in and show you. So this was more of the summary of the data that you were looking at in the cluster. So when I zoomed in one of those, because I first wanted to check whether this approach works, because what I'm doing is just mixing them and then see if biologically it's making sense. So what you see here is just I took one of the plots here is where I see that when you in the placebo arm, you see here a higher expression of one cell type, and when you go down to the combination therapy, that signal is not there. So what this tells me is, and further I identified what these populations are because I have all these infos here. So these are the CD23 positive cells on the CD14 cell population, so CD23 expressions on monocytes. And then later on I found out, So what I found out that there are evidences where it shows that in case of allergy, there's a higher expression, and then when you give the therapy, then it goes down. So this is just in, like I was trying to chase the fact first, that what's known out there. But my motive here is that if we can find out some novel cell populations, because these are known, right? But by this approach, if we can correlate the cell population which correlates with an endpoint and we don't know anything about it, that would be something interesting to look after. So this approach can help you to identify the novel cell populations. And so let me change my gear and say talk a little bit about more on. So what I was getting to earlier was that what you could do with this import data. So you could download import data, and one of the things that we are interested is to understand the normals. So on import side, we have lots of flow data available, and so we separated out the flow data, the control sets. So the data sets where we have control information, we put all those together, and then we're trying to normalize, because normalize was the first one, because when you have different studies, different labs, it's very difficult to normalize the data. So we wanted to make sure that they are all in same level, and so to do that, so we downloaded about by now we have about 20,000 flow files. And what we have done is, and these are all from the control set, and what I'm showing here is the raw data for the CD4 channel, and we normalize the data here, and if you can see here that these are, we are trying to identify the peaks or the mode of the distribution, and the color is based on different studies. So within study they align pretty well, but across studies you do see variations, right? But when you want to set up, you want to come out with a reference set of normals, then what you do is you need to do you have to normalize all them across studies and also within study. So within study and across studies, what you see here is now all these peaks are aligning. If you want to know more about this normalization, this paper came out in 2010. It's a per channel basis normalization method, and that came out from Brinkman group. And so the peaks are here called landmark identifications. So they identify the landmarks. For example, here are 2 landmarks, the modes, and then they did a very simple k-means clustering approach. So cluster all the landmarks together, and then they aligned it and normalized the data. So this is just the one step towards how we can now put together all the normal data. Which itself is challenging, but this is the step that we took. That's the first step. We have much more to do. Then the other thing that we are working on is more of the text mining approach, the literature mining. So what's out there? Again, like you can at the most read in a day 5 or 10 papers, but this kind of approach really helps you to get information from the literature. So this is more of a question that we are asking based on cytokines. So so what you see here is that we got about 44,000 abstracts based on the mesh terms of what's the likelihood of co-occurrence of the disease, cytokine, or cell type. So we got 44,000 and then separated them out based on these rules of if there is a disease cell and a cytokine, they exist together, and then disease and cell combination, disease cytokine, cytokine cell, and number of different co-occurrences. So that all led us to about identifying about 1,500 disease, 65 cell types, and 84 cytokines. So this work is done by Nofar Giffman. He's a postdoc. She's a postdoc in Butte Lab, and then she came out with looking at these occurrences based on the disease. So, for example, what I'm showing here is that you want to understand how these cytokines and cells, they are involved in diabetes. So, if you see here, these are the in green are all the cytokines, and in red are the cell types and how they are connected. So these are the starting point for generating a hypothesis and finding out, okay, what are the ones where you should be studying, or at least give you the pointers. We are still working on the edges right now to define, because that's the most difficult part, but these are only based on co-occurrences of the disease, cytokine, and cell types. And this is more of cytokine centric, where let's take one particular cytokine and see in how many diseases they are involved, and the size of the node is proportional to the number of abstracts where they found the co-occurrence of the disease and the cytokine. Okay, so I don't know how much time I have, but this is my last slide on this is another way of getting the data. And nowadays, you know, the Facebook, Twitter, and news reports, you can also get all this information from these sources. And how you put all this together is like, for example, you know, I'm going to show you just the website where, you know, people have this was one of these examples where they had cholera related tweets which tracked the early stages of 2010 cholera outbreak, and like that there are many, like I'm hearing a lot about Ebola virus right now. So if you go to this site, let's see if it works. It's called healthmap.org, and you know, it's giving you like the, let's look at the world map first. So it's giving you like there are 171 respiratory alerts based on all these Facebook, Twitter, and other resources. This is just most the fun part. And then this also lists you where it is, and you can select what disease you want to look at. So for example, if you're interested in influenza, you can select influenza. This is just showing US. You can set up under location, you say all locations, submit the search. So then it will do a worldwide search. Hopefully it's going to work. So these are different. So in the month of, say, January, there was a peak, and this is like in the United States, and you can see other countries. And so this is another way of how the information is getting captured. And with that, okay. So my take-home points are that, you know, the pattern-based visualizations are a very effective method. Combinatorial approach can help you identifying the novel populations. from the minimal set of markers which are needed to characterize functionally distinct cell populations which are associated with clinical outcome. So that's my next step, that I want to come out with some minimal set of cell markers which will help you to characterize the cell populations which are associated with clinical outcome. And while individual studies are great, but when you can pull the studies or do meta-analysis from the public repositories, they are quite powerful. And then knowledge-based data-driven techniques. And with that, I would like to just go for the acknowledgements. Atul, he's leading the input project, and then Sandra, Jenna, Nofar, and some of the slides I've shown is their work. And this is in collaboration with Northrop Grumman, who are developing the database for us, and is funded by NIAID. And we have ontology folks on board as well. So, you know, it's very important when you're curating the data, you need to have controlled vocabularies. You just cannot work with free text. And that's something is very, very crucial for curating data. So our Buffalo team is Barry Smith, Alan, and Alex. We are working on ontologies with them. And with that, I would like to thank everyone. And if you have any questions, feel free.
Questions for Sanchita? Everybody ready for lunch? Okay, well, thanks very much. And that brings us to the end of our morning lectures. As you probably realize, the afternoon is a little bit different today from the first 2 days. What we have is a set of demo stations. There will be 2 of them in the basement of CCSR. There's some small seminar rooms there. Is Mina still here? Yeah, there. Mina will be guiding groups between the rooms in CCSR and the Protein Simple or NanoPro demo, which will take place in the Grant building. So you've been assigned actually to a group, so you'll see that in your binders, your handout. And Mina and Angelica will shuttle you to the right rooms at the right times. Basically, that just gives us opportunity to have a small group to do a more interactive kind of discussion / demo of one will be Luminex, one will be Stanford Data Miner and other data mining techniques, and the third one will be, what is the third one? The Protein Simple nanoimmunoassay. So essentially it's an hour at each station, you rotate, and that'll be our afternoon. Lunch is the same place as usual in CCSR, so we can break, and they actually, I think, are just